This video will discuss the California Condor Microbiome Research Project and how the results of this non-invasive research can help attorneys prove harm as required by the Endangered Species Act. The California Condor Microbiome Research Project is being led by Dr. Jonathan Longmire. Dr. Longmire has his PhD in genomics and microbiology and is a senior advisor at the Los Alamos National Laboratory in the Biosciences Division. Dr. Longmire was kind enough to let me interview him to discuss the work he's doing with condors. In addition to Dr. Longmire, I was able to interview Professor Dan Rolfe from Lewis and Clark College of Law. Professor Rolfe is an expert in environmental law, wildlife law, and in particular the Endangered Species Act. A microbiome is the ecological community of commensal, symbiotic, and pathogenic microorganisms that literally share the body space of an animal. This term was originally coined by Joshua Lederberg. He argued the importance of microorganisms inhabiting the body and how that affects health and disease. In other words, all of the microbes and bacteria in the body of an animal is an overall health indicator of that animal. The healthier the microbiome, the healthier the animal. Microbiomes can address indicators such as nutrient levels in the animal and reproductive health in the animal.
The California condor is one of the world's rarest birds. Today, there are only about 300 of them in existence. At one time, they may have ranged across much of the continent. There's evidence that they ranged west of the Rocky Mountains from British Columbia to Baja and across the southern U.S. to Florida, and there are even fossils that place them in New York State. Due to the settlement of the country, their habitat slowly shrank until by the 1940s they were limited to 6 counties in California just north of Los Angeles. By 1987 there were only 22 birds in the world, all congregated in the mountains above Santa Barbara. These birds were captured and placed in a captive breeding program. Today, the number has increased to just over 300 birds. About half of them are free-flying, with the remainder remaining in the captive breeding program. The free-flying condors are in 3 populations in California, Baja, and northern Arizona. The condors that spend the summers in southern Utah are from the northern Arizona flock. Condors are one of the largest flying birds in North America. Their body can be from 45 to 55 inches long. They may weigh more than 20 pounds with a wingspan of over 9 feet. The young of the year have dull black feathers with a dull black or gray head with no feathers at all. As they mature, the skin of the head gradually turns yellow, orange, and red. The transition is complete in about 7 to 8 years. These birds are strict carrion eaters. They use their great wings to soar effortlessly for hours in search of food. Condors mate for life and normally nest only every other year. Eggs are laid directly on the ground, usually in a cave or large overhang on an expansive cliff. They hatch in about 56 days, and the young birds fly about 5 or 6 months later. The parents tend the young birds for more than a year from fledging. The Kolob site in southern Utah probably represents the best opportunity to view California condors in the wild. Over 30 birds have been seen frequently near the road just 4 miles south of Kolob Reservoir. The future certainly looks better for the condor today than it did in the late 80s. They still walk a very thin line as they face survival challenges from such things as predation and lead poisoning. Hopefully the future will continue to brighten for this magnificent vulture through the efforts of federal and state wildlife agencies and private interests such as the Peregrine Fund.
The California Condor Microbiome Project will compare the microbiomes of condors in the wild with condors in captivity. The hypothesis of the experiment is that the condors in the wild will have healthier microbiomes than those of the condors in captivity. The reason for this hypothesis is that the condors in the wild have access to a more varied diet. Wild condors inhabit large territories, often traveling 160 miles a day in search of carrion. It is thought that in the early days of its existence, the California condors lived off the carcasses of megafauna which are now extinct in North America. They still prefer to feast on large terrestrial mammalian carcasses such as deer, goats, sheep, donkeys, horses, pigs, cougars, bears, or cattle. Alternatively, they may feed on the bodies of smaller mammals such as rabbits or coyotes, aquatic mammals such as whales and California sea lions or salmon. Bird and reptile carcasses, however, are rarely eaten. Alternatively, condors in captivity are fed a diet of only quail meat. The varied diet exposes the wild condors to different microbes and pathogens, thus leading to a healthier microbiome. The microbiome is a scientifically proven method to show the overall health of an animal, including the reproductive health. The experiment is done by obtaining fecal matter samples from both wild and captive condors. The fecal matter is tested in the lab using DNA sequencing technology made available by the Human Genome Project. This method of data testing is non-invasive to the condors and can be replicated over and over again in the lab. The Endangered Species Act of 1973, signed into law by President Richard Nixon on December 28, 1973, was designed to protect critically imperiled species from extinction as a consequence of economic growth and development untempered by adequate concern and conservation. The U.S. Supreme Court found that the plain intent of Congress in enacting the ESA was to halt and reverse the trend towards species extinction, whatever the cost. Under the ESA, it is illegal to harass, harm, pursue, hunt, shoot, wound, kill, trap, capture, or collect, or to attempt to engage in any such conduct. This provision applies to captive species as well as species in the wild. The definition of the term harm under the ESA is very broad. In Babbitt v. Sweet Home chapter of Communities for a Great Oregon, the United States Supreme Court defined harm as an act which actually kills or injures wildlife. Such an act may include significant habitat modification or degradation, or it actually injures or kills wildlife by significantly impairing essential behavioral patterns, including feeding, breeding, or sheltering. Section 7 of the ESA applies to federal action, and Section 9 applies to private action. Section 9 has a citizen suit provision, which makes it less difficult to bring a successful claim under the ESA than it is under the Animal Welfare Act. Although the definition of harm is very broad under the ESA, proving actual harm is a difficult, time-consuming, and expensive process. Often a suit will not be initiated because of these difficulties. Having reliable science showing that an animal in captivity is not as healthy as its counterpart in the wild could be a very useful tool in proving harm under the ESA. And because the research being done on condors is non-invasive, I would suggest that it be expanded to other animals such as elephants to show that captivity is harmful to them as well. Using a scientific approach to proving harm to an animal is a very useful tool, and in particular under the ESA, which has a citizen suit provision. I think that this project could have positive implications for progressing the cause of animal welfare.